leads to autism. We didn't know that part. They went together, but we didn't know that part. Now, more recently, we've been using these fancy techniques where instead of looking at the chromosomes and being able to resolve maybe 1,000 different marks on the chromosome, we're using these microarrays that allow us to resolve 1,000,000 spots along the chromosome. And using these Affymetrix arrays, we can now recognize that these copy number variants are extremely common in autism, and this is now generally recognized in the literature. It's not found in controls. It was found first in patients with autism. It's now recognized that the same kinds of copy number variants are increased in bipolar disease as well as in schizophrenia. And we don't still understand the exact mechanism that underlies these copy number variants. But I want to show you what they look like. This is, instead of looking at the chromosome as a cartoon here, this is the fancy way that we now look at them with the microarrays. The chromosome is laid on its side. This is that same chromosome 15 that I was showing you. And this is what we call a heat map. And what we have on each of the vertical lines is a piece of DNA sequence that we're evaluating. And there are thousands of these sequences. I don't know if you can see the striations here. Each horizontal line is a different child's chromosome that's been laid along that same axis. And we're reading that sequence across. The intensity of the color tells you how many copies of that DNA there are. So the average color is this orange color, which says there's 2 copies, one from mom, one from dad. At this yellow end, we're having 4 copies, and at the red end, we're having 1 copy. So these are those kids where I showed you that 15q inverted duplication. They have a dose of 4. Here's a child that has an interstitial duplication with a dose of 3. And here's a child in that same region that has a deletion. So this is a surprise. These things weren't known to exist very long ago. And lo and behold, we've got them not only existing, but they're clustering in certain specific regions on the genome. This is another deletion nearby, but it's not affecting the same genes. If we go to a higher resolution and look at that, I'm not sure that you can see the axis, but basically this is showing the 4 copies dropping to 3, dropping to 2. And these are those duplications. And what we're able to lay them over are the genes that are impacted. Now, we don't yet know the relevant gene. It's a relatively large block. It carries about 20 million base pairs. But there are a number of interesting candidate genes in neuroreceptor metabolism that are there that are intriguing candidates to look at. Now, there were hints of these kind of energy deficiencies. I said they were showing you the biochemical markers. And the question we asked is that causally involved? Is that an important part of the syndrome? Well, we then went to a large population. This was with Pauline Philippek and Moira Smith. We were looking at a large population that had normal karyotypes. They didn't have these unusual chromosomes. We wanted to see, did they have the same biochemical signal that we saw in these unusual individuals? And yes, in fact, they did. But you couldn't see it taking one at a time. You had to take the whole cohort of 100 and compare them to a cohort of 100 normal. And you could see at a very high statistical probability, at a p.001, 1 chance in 1,000 that this was just by chance alone. So we could see this same signature reflected some subset of kids who did not have the chromosomal abnormality, suggesting this may in fact be a component of what's going on in autism. After our large retrospective study that we published in Journal of Autism and Developmental Disabilities in 2004, a follow-on study from Portugal was a population-wide study where they found the same biochemical effects that we had seen in our study, but they followed it on by actually doing biopsies and measuring mitochondrial function and looking for mitochondrial DNA mutations and not finding the mutations. Again, suggesting that's what's going on is a normal chromosomal DNA a defect in mitochondrial function that's found in a significant component. From their studies, they estimated that maybe 5 to 7% of the kids with autism were affected with this biochemical signal. Now, I mentioned that we had another major energy-producing pathway, fatty acid oxidation, that actually was the lead-in for our group here at UCI to become interested in this correlation a long time back in 97, when we saw abnormalities in fatty acid oxidation in some very unusual families. And The fatty acid oxidation signature is carnitine deficiency. That's the signature finding. And carnitine is the little shuttle molecule. It's a vitamin that brings the fats into the mitochondria and allows them to be burned. This class of diseases is a very new family of diseases. It's only about 10 years old. There's about 21 of them, but they have a very major impact on child health. They play a major role in SIDS, or early infant deaths. And it really was the reason, these family of diseases were the reason that the newborn screening test has been expanded out to include the large number of tests that are now being tested. We used to test for only 4 or 5 diseases. Now we're testing for several dozen. But the push came to find these diseases that cause this very sudden infant death syndrome. Now, this goes back to the early notions that we had that there may be a role for mitochondrial defects. It was a study that I had done with Mark Lerner, where there was a family that had 3 children with autism spectrum disorder, and the eldest had died a SIDS-like death. So we started working up the family for these fatty acid oxidation diseases. A second child had a near SIDS encounter, and they all had hypotonia and the signature biochemical signals that I had mentioned to you earlier. And we were able to work them up and see that there was a block in fatty acid oxidation and the signature of mitochondrial proliferation, even though the respiratory complexes in the DNA were normal. Again, saying that there's functional defects in how the mitochondria are working in these very rare forms. Now, this carnitine deficiency is actually an intriguing insight because of the fact that valproic acid, a seizure medication that's given to children, causes a major risk for carnitine depletion. In fact, nowadays when kids are started on the drug, they're always measured for their carnitine levels before the drug is initiated. It turns out that this drug that causes carnitine deficiency is really the best teratogenic model. Teratogenic means you expose mom to the drug and the baby carried in utero can come down with the syndrome. It's something that's been seen very clearly in humans in a very narrow window of exposures early in pregnancy, but it's been very well studied carefully in models in mice. So it's pretty clear that there's something involved here that can cause behavioral phenotypes that look very much like classic autism. Now, those of you who are putting money into the bank realize that the bank account can be low because you're not putting it in fast enough or because you're taking it out too fast. And so that leads to another related class of inborn errors, of genetic diseases that we see that cause what I call an indirect energy deficiency. And this is a set of neurotransmitter diseases that I only show very quickly in passing because it shows how identifying a target gene allows you to intervene and carry out therapy. This was a child sent to me for evaluation because they thought he had mitochondrial disease because of these signature findings in the midbrain and the characteristic phenotype of severe delays and ataxia and seizures. In working the child up, we were able to identify that he had a very rare genetic lesion called SSADH. I'm not going to give you the full name, but the shortcut of the name is GABA hydroxybutyric acid, gamma hydroxybutyric acid. It's the date rape drug. The genetic block was causing the kid to make his own date rape drug. There have only been 4 kids reported with this, but a mouse model had been made of this syndrome. A knockout mouse had been made. Now, the knockout mouse died quite quickly, but it stayed alive as long as they could make it eat breast milk. And they were curious, what was it about breast milk that was keeping them alive? And they found that the breast milk was very high in taurine, and in fact, they could keep the mouse alive as long as they were forcing it to eat taurine. Well, as I said, there had only been 4 previous kids with this disorder, and when I shared this information with the family, they elected to feed the kid a truckload of taurine. Sure enough, in 1 year of treatment, his development improved. We cleared up the lesion in his brain, his ataxia resolved, and he still has seizures. It's not that we fixed the situation, but it's had a pretty dramatic impact. But it's a therapy you never would have dreamed of without having targeted a specific gene. Again, the importance of being able to try to resolve things down to a specific genetic focus. The last of these types of indirect indeficiencies are the ion channel diseases, again, that I mentioned briefly in talking about the